let's take a look at an example of how we can use visualization and actually take a first quick look and actually develop a much more complex representation. This is some work we did here at Caltech with the Barbara Wald lab on expression of mouse genes as they develop from mouse embryo muscle into specific types of muscle. So originally what they had is they had a genome sequence and they were able to measure the strength of bonds between different parts of the genome. So the way they visualized this originally was using a product called GraphBiz, which is actually quite an old package that came out of AT&T labs, Lucent labs, back in the 80s called Dotty, maybe even further back than that, which is basically a system to visualize graphs. Now, the strength of Dotty is that if you have many connections, not only can you adjust the color and the width, but it'll make an effort to make sure that the lines don't intersect each other. Now, while this was good for a first quick inspection, this is not necessarily what they needed to see. So a first good place is to actually see what's out there. What else can they take advantage of? Here's a visualization from what's called the map of science. About 800,000 published papers were analyzed, and they tried to see what connections, what were being done between different fields. In other words, what papers were being referenced from one field to another. They used that in a clustering system to actually bring the fields together and actually represent the papers and represent these connections through lines. So you can see very clearly that in this case, brain research is very close to computational science, as is in general astrophysics and math. You can also see there's actually the different papers actually being shown as line of text coming out of these different areas or different clusters. While this is a very complex and interesting visualization, it is not very easy to kind of inspect and analyze and kind of look at very specific information. So some work was actually done to make an interactive version of it. And they actually make it as a ring plot with the different connections actually not only done the different connections not only done as arches, but they use what's called edge bundling to make connections that kind of are close to each other kind of be bundled into connections that actually make visual elements. This was some work done by Eigenfactor, and it is fully interactive. You can go to a specific journal, you can click on it, and it highlights those connections. Notice that this might be very similar to what the biologists wanted to see when they wanted to see whether certain genes were very special or important to actual gene expression. Here we have a zoom in of a particular region, and you can see the very specific connections between different journals. Notice that each of these connections would actually be a paper referencing one area to the other. Now, in the area of biology itself, there has been lots of work to actually visualize complex systems and kind of find ways of summarizing it. So here we're looking at connections between different chromosomes and RNAs, and simplified areas that get actually drawn with this kind of squiggly lines, and bonds are actually shown with this kind of directional graph. This was called the ABS Explorer that came out of Vancouver Cancer Research. Now, Ben Fry, who we mentioned before, has actually also worked on gene visualization in general. He actually did some work to actually visualize a gene for comparing different animals, different species. And he came up with a system which actually became a fully interactive 3D environment where you could dive into the information. Well, as it turns out, any user that worked on this became seasick really fast, motion sick. And he realized that what he needed was something simpler and cleaner to let them look into the data. So he came up with this CD36 browser, which basically has different levels of details encoded as different layers of information. You have the top view on top, and the top part of this plot, with actual markers where interesting information, what the big differences are between the two animals. And you can zoom into the sections, and the closer you can zoom, you eventually can get to the genetic code. So how did we put it together to come up with our solution and come up with all these different solutions to actually come up with something useful for our researchers? Well, the idea of these connections and the interaction to actually be able to highlight connection became very clear that was very important and that would be very useful for researchers. So we created an interactive tool that actually lets us mouse over a specific gene and actually shows the connection to different genes. Not only that, but actually shows the strength of the connections. It shows detail in the particular genes. It even shows the signals that are actually able to mark for each of these gene sections. In this case, the biologists already had this protocol that important genes connecting to other important genes can be isolated, so we actually use those connections by mapping them on the bottom rather than on top. Now, while this became very useful for looking at small subgraphs, it turns out that what they really want to do is they want to be able to do this massive research projects and do these experiments and then be able to push this output into visualizations that they can explore. It turns out that our tool that we created could very easily be manipulated to become more stable and actually dump out full plots that could still be semi-interactive and searchable. So what we did is we actually created SVGs, which is equivalent to a PDF, but in XML format. And we were actually able to visualize the whole genome at once. Here is actually the results. Obviously, you can't see anything because it's just one, looks like a very thin line of white in the middle of the screen. But the truth is that if you actually show this if you actually printed this, it would actually be 1 foot high by 1 mile long. Now, if we actually start zooming into it, then eventually start looking into the detail. And the reason we're showing this is because the SVG resulting from this is actually something that a web browser can handle. You can actually bring this 1 foot wide, 1 foot tall by 1 mile wide plot into an SVG, and you can search for any different label, and it'll take you to the part of the genome sequence that you're looking for. Now, this is actually something that was made to be part of their research environment, so they can use it regularly. But we even took it one further step and said, okay, what other representations can we find? In fact, we developed an art piece, which is more of a concept, which is actually representing these sequences rather than linearly put them in a spiral. This allows us to put a whole chromosome in one single spiral that then can be printed in a much smaller space. Not only that, but if you actually printed this in some kind of semi-transparent media, like vellum, you could actually print the same data set before specialization, after specialization, and you can lay it on top of each other to analyze the differences. We actually printed these as a complete sequence, and we were able to create an art piece that actually shows the three complete gene sequences for the data before specialization, after specialization, and after specialization with an inhibitor. And as it turns out, that it actually became quite useful. If you walk around this system, it does all the things that interesting visualization can do. It shows you data problems. There were some genes some chromosomes that are actually showing much more information than they should have. It showed the differences. You could see particular subgraphs that were actually being very active before and not afterwards because of an inhibitor. And you could even isolate the particular genes that were interesting to the researchers because they knew where to look within each of the chromosomes. So let me conclude and kind of give a whole summary of what we've talked about. And that is that scientific visualization is one of the big keys to understanding science. Not only that, but the way we approach it and present it can be quite powerful. And if we know how to do it, we can actually show a lot of information and make it discernible and really use well the cognitive system that a human comes with. However, the specifics of how to put each of these things together really comes down to what is your goal and having a good understanding and going through this iterative process of coming up with the best solution possible. So this quote that Colin Ware got from a review of a cognition system is actually quite strong, saying that we are truly an artisanship of how to put these different sciences and principles and concepts together, But if we put it together, we can really accomplish strong things that are actually quite insightful. Thank you.